The aim of this protocol is to identify a method which can be used for identifying novel genes which are involved in calcium signaling. To do this, we use the classical forward genetic screen. Calcium signaling is an important defense response pathway in several plant systems. In order to identify the genes involved in this pathway, we have used the classical forward genetic screen. In this method, EMS would be used as an alkylating agent to introduce random mutations in the plant systems. The developed mutant generation can then be used for screening for a phenotype of interest. Once the phenotype of interest has been identified, the causal gene can be mapped. In this study, we use the model plant Arabidopsis, which has a calcium reporter Aequorin in its background.
Weigh 150 mg seeds of Aequorin for EMS mutagenesis and another 150 mg seeds to be used as control. Transfer the seeds to a 50 ml Falcon tube and add 0.2% EMS or autoclaved water. While using EMS, be careful because it is carcinogenic. Protective gears should be worn while using EMS and any kind of spillage should be prevented. Seal the Falcon tubes with parafilm and wrap it in aluminium foil. Rotate the tube end over end for 18 hours at room temperature. Allow the seeds to settle and remove the EMS solution carefully and discard in a waste container containing 1 molar NaOH. Wash the mutagenized seeds thoroughly with 40 ml of autoclaved water at least 8 times. For the final wash, add 100 millimolar sodium thiosulfate and rinse for at least 3 times.